frequently frequently we use rodents that we are able to make genetic changes on them, introduce different pharmacological drugs, or make transplant in that animals. They are another species that is important for in vivo studies as worm or zebrafish that contribute to mostly to understand the neurodevelopmental biology and also a basic like acknowledgement about neurology. If we are going in deep in the in vitro models, we know that cell culture, help us to understand the cell-to-cell interactions. Is a simple manipulation, and the cell culture offers a single microenvironment to analyze basic conditions in the cells. This classic in vivo model platform, mostly we use rodents as mice and rats, fish, fly, or human tissue. We can obtain cells from these organisms and make immortal cells, which is the cell lines or primary cultures. In the last decades, these models have helped us to understand a lot of mechanisms of neurological disorders. However, these cell cultures have been some disadvantage, as like of physiological complexity, static condition like a physical and biochemical signaling, large region values, uniform concentration of nutrients and drugs, change of phenotypes of cells, change of gene expression. When we are using different species compared with humans, we have a different genes profile. There are changes in morphology and polarity and limited predictability. For that reasons, about 20 years ago was introduced a technique which is named reprogramming cells. Since that we need to try to mimic as possible the human condition in neurodegenerative diseases. So we have in this case, this iPS-based models is based in take somatic cells from a patient and use specific transcription factors related with embryonic stage. We force to these cells to express the epigenetic, genetic, and phenotypic characteristics of embryonic stem cells, that is named induced pluripotent stem cells. These cells are able to differentiate mostly all kinds of cells of the human body, or they are able to produce cells from the three germ layers, embryonic layers, as mesoderm, endoderm, and ectoderm. These cells or this kind of technique have helped us to develop cell therapy models, drug discovery, therapeutic screening, and in vitro model diseases. Is the case in neurodegenerative model, we can obtain cells from a patient with Alzheimer's disease. We can reprogram the cells to a pluripotential stage or a pluripotential phenotype and differentiate to different neural cells as neurons, astrocyte, microglia, and understand or discover molecular pathways and novel disease mechanisms specifically from this patient. However, this is a 2D condition, getting the same disadvantage that I explained before. For that reason, in the last 10 years, we start to develop 3D in vitro models or organoids. Organoids is defined as a cellular system that emerged by self-organization, means that pluripotential stem cells or induced pluripotential stem cells in the genetic in their genome have the morphogenes information, means that the cells in a matrix are able to organize for itself given a tissue organization depend of the growth factors that we induced. When we produce organoids in this matrix, or these cells are differentiate and divide in multiple cell types and exhibit some cytoarchitectural and functional features, reminiscent of an organ or region. Organoids can be generated as a 3D cultures or combination with 2D culture. This is also named co-cultures or assembloid as well. These organoids are able to survive in culture for long periods. The last studies have shown that a brain organoid we can measure for 1 year. For nervous system, organoids are generally constructed from pluripotent stem cells. However, they are a model for tumoral organoids, so we can obtain from tumoral cells and also some adult stem cells. Specific mutations can be introduced into organoids to study their effects in neurodevelopment. And the study a specific disease in brain organoids can be generated for individual patients, thereby preserving the specific genetic background of the individual and generate an insightful model. That means as cells conserve the genetic background, we can mimic or modeling in the organoids the characteristics of some specific disease, mostly when we are working with familiar mutations. If we compare the 2D versus 3D cell culture model, we have that in 2D, we have a diffusion of soluble factors into the media. They are not gradients. The cells are exposed to liquid interface and they are forced to cellular organizational and polarity, which is different in vivo. They are a restricted cell-cell interaction and the high stiffness promotes monolayer splitting of cells. In 3D conditions, the cells are embedding in a matrix or in a scaffold where the gradient, the soluble factors are released in the organoids as a gradient, a concentration of gradient, more similar to in vivo conditions. The cells adhere to extracellular matrix via integrin matrix binding, and the matrix fibrils restrict the cell splitting and they are more cell-cell interactions. This is a picture, this is how looks a cerebral organoid in a draw, and here how looks we can mimic some areas of the brain. For that reasons, organoids can bridge patient and animal study to advance our understanding in neurological diseases. Classically, the patient study start mostly when the symptoms onset or when they are a diagnosis. However, the initiation of this disease is unknown, so that produce like a difficult to start studies in patients. In animal models, we can control the disease initiation, we can follow the pathogenesis and symptom onset, and like make a timeline of this course. However, animals, the genetic profiles and transcriptomic of rodents mostly is not similar to humans. And that is the reason that when we try to translate this size to human, in the cases not works. So organoids could be, can be a bridge between clinical studies and animal models to know and transfer, release, and conserve these disease mechanisms. What is the classification of neural or cerebral organoids? We can classify, when we differentiate these IPS into the cultures, we can get neurons, astrocyte, microglia put in an organ-on-a-chip preparation, or we can get 3D cultures and produce unguided neural organoids. One means unguided, means that we can get different areas of the brain in the same organoid, or produce guidance organoids or regional organoids, means that we can differentiate in specific areas of the brain. For example, spinal organoids, striatum, and cerebral cortex. When we fuse these organoids, we can get assembloids, and we can make interfaces, or we can make connections between these organoids and makes it functional. Another option that we have is get these organoids and transplant in rodents models, and some the murines help to give us some cells that we are not able to produce in the cerebral organoid models. So in the last years, there are many publications that have shown different kind of regional organoids. So we can get the whole brain, cortex, basal ganglia, the hippocampus, striatum, retina, thalamus, hypothalamus, midbrain, cerebellum, and spinal cord. Mostly, the cerebral organoid models cover the most part of the brain. Their advantage and disadvantage depend of the kind of the model. I will explain after, but mostly it's talking about variability. When we have an unguided organoids, we have more variability in the model than regional. What is the mainly characteristics of the brain organoids? So we have first the colonies. This is the IPS colonies after reprogramming. Then we can obtain the embryo body, which mimic like the embryo stage, and then the neuroectoderm. Neuroectoderm mimics the neural tube in the neurodevelopment. So here in this image, you can see on this draw, you can see the neuroepithelia. The neuroepithelia is the area when, or the neural tube where we can find neural stem cells and radial glia. These cells in the neurodevelopment are in charge of the production of different kind of neurons and glia cells during that neurodevelopment. When the organoid start to measure, we are able to produce in vitro different areas of the brain. The most important one is the ventricular and subventricular zones, the outside ventricular zone, deep layer neurons, and upper layer neurons mimic some neocortex area. This is very important because if we compare with mice or with rats, these murine animals, they are not able to develop the outside ventricular zones. And this area in neurodevelopment is very important for neocortex in humans. So this is how looks the organoids after the first stage of maturation. We can detect these subventricular zones and the neuroepithelia using specific markers as SOX2, and then we can see using specific markers to detect cells from deep layer neurons and upper layer neurons, how to the organoids start to organize as a human brain. And finally, after 120 days, we can start to see glia cells or astrocytes that start to make interactions with neurons. Around 120 and 180 days, this is how looks the organoids. So they can reach sizes from 0.5 millimeters to 1 centimeter. And if we analyze the histology using immunofluorescence, we can detect areas of the brain in these organoids. For example, that is the cortex, the cortical areas in the organoids, when we can identify different layers of the human brain and which we can find different kind of cells or neural cells. Neurons as pyramidal neurons, glutamatergic, GABAergic, dopaminergic, depending of the kind of the organoid. What is the applications? Mostly is investigate the causal relationship between risk variants and define cellular phenotypes. Organoids can replicate patient-specific genomic background, study the mechanisms underlines the development of the disease with a known cause, dissect the mechanisms of pathogenic mutation in human tissue, and screen potential therapeutic targets. Mostly, the literature in the last year have published using human organoid comparison between interspecies and understand the human evolution. Also, congenital brain malformation, the major psychiatry disorders, we can make a screening, drugs screening in these organoids, and neurodegenerative disease when we induce aging in the organoids. Today, I will show some studies that I have participated, mostly using organoids as a model of neurodegenerative diseases. The first model that I participate is modeling of amyloid beta and tau pathology in human cerebral organoids. Briefly, the neuropathological features of Alzheimer's disease, which is part of amyloid beta and tau pathology, is alpha secretase and beta secretase they are a produce of accumulation of these proteins, producing the plaques and neurofibrillary tangles when they are a hyperphosphorylation of tau that produce neuron loss and degeneration that makes or call the pro-inflammatory factors in the brain. This role is make as with microglia is active in this case, and they are a blood-brain barrier leakage and neuroinflammation. So that we made is develop a cerebral organoid from a familiar Alzheimer patient and Down syndrome patient. Since Down syndrome, the chromosome 21 is related with a ApoE4 gene, which have a relationship with A beta and tau pathologies. So we found in this model, using specific markers for A beta plaques and tangles, we developed after 120 days of cultures that these FAD organoids, so familiar Alzheimer organoids, and Down syndrome organoids were able to develop this is histology characteristics of that disease and increase mostly in Down syndrome compared with FAD. Also using markers for tau phosphorylation as AT8 and PHF1, we found the hyperphosphorylation of tau in that organoids in both FAD and Down syndrome compared with the healthy controls, with healthy organoids, cerebral organoids. Using Western blot and ELISA, we analyzed also that the levels on insoluble tau and total tau, as well as phosphorylation of tau, increase in FAD organoids and Down syndrome organoids. Also, as a control, we used reprogramming cells from a patient with a Cross-Jacob disease, which is a disease related mostly with prions that is not they not produce a beta amyloid and tangle, so we didn't see the activity of phosphorylation of tau. And finally, we analyzed the levels of caspase-3 cleavate. Caspase-3 is a protein related with apoptosis. So we saw that in FAD organoids and Down syndrome, the levels of caspase-3 activated increase compared with healthy controls. So we conclude that we can use we can take samples from a patient, familial AD and Down syndrome patient, develop cerebral organoids, and after 2 months, we can obtain A beta amyloid plaques. Also, there are another study after this one that show that using sporadic samples from sporadic AD patients, we can obtain as well A beta amyloid plaques. It was the first study using organoids as a model for Alzheimer's. However, nowadays, there are more robust models using another kind of cells and make a stronger screening, a cell single transcriptomic, and have shown that this model mimics well that disease. Another model that I have participated is the traumatic brain injury in organoids. So briefly, traumatic brain injury is a trauma produced mostly for car accidents or for some sports as a football or boxing that produce a trauma in the brain, generate a brain barrier breakdown and edema. There are gliosis and infiltration, and there are neural death and tissue damage. And this, with the time, this kind of trauma also is a risk factor for neurodegenerative diseases or dementia. So usually we use mouse brain to mimic the trauma put the brain of the rodents in a compression, in a different compression, and then we check what is the level of the trauma in the brain. In this case, we design a phantom brain. Phantom brain means that we use the skull of the mice, and then we introduce a human brain into the skull using agarose, and then we put this brain in a different compression, and we analyze if this organoid produce some characteristics of the trauma. Initially, we characterize this organoid. We need to be sure that the cerebral organoid are able to generate neuroepithelia and the different layers of the cortical brain, human brain. We compare this phantom brain with the classic traumatic brain injury model, and we saw that organoids present increase of GFAP levels, means they are astrogliosis. This is a signal of cellular stress in the brain, because gliosis means that they are pro-inflammatory factors in the brain. So we saw, compared with the traumatic brain injury in a mice model, that there are increase of GFAP as a mouse brain, and also there are a decrease of MAP2 levels. MAP2 is a marker for neurons. Here we analyze that there are elevated neural damage in cerebral organoid after compression, very similar to the traumatic brain injury model, and using specific dyes for detect apoptosis and CAS phase 3 also, we saw that they are elevated reactivity of these markers, very similar to the mouse model. So the only disadvantage of this model is we don't have microglia or the cells that is related when the immune response, and this is important. So we decide to include in the future for this model this kind of cells to try to mimic as possible the human brain. This model is using regional organoids. I developed this model, this Lewy body-like pathology and loss of dopaminergic neurons in midbrain organoids. So in this case, we try to mimic the Lewy body pathology in the midbrain. So Lewy body pathology is due to in the midbrain, dopaminergic neurons have an important protein, which is the alpha-synuclein. Alpha-synuclein is involved in the functional of dopaminergic neurons, and dopaminergic in the midbrain is in charge of some movements, or is in charge of the movement functions in the brain. That is the reason that Parkinson's disease is related with this physiopathology. So when we have an aggregate of alpha-synuclein, we can get oligomers and then protofibril oligomers and fibril aggregates, producing a Lewy bodies. Lewy body are intracellular in the neuron and produce the cell death. In this case, we use samples from patients with a mutation in alpha-synuclein. The gene that codifies for this protein is SNCA. So we could develop a triple mutation for CNCA and then generate this midbrain organoid. So you can see here the human IPS, the embryo body, then the differentiation to neuroectoderm, the neuroepithelia, and then midbrain organoids. In this case, we use specific markers to find the neuroepithelia in the beginning, and then markers specifically for midbrain areas. And finally, markers for dopaminergic neuron, which is involved in the pathology of that disease. And after 180 days, we found some phosphorylation of alpha-synuclein, mostly a hyperphosphorylation, intracellular phosphorylation, and then aggregations inside the dopaminergic neurons. If we compare with the human brain, we found the histological characteristics of the Lewy body formation. This is how looks the Lewy body in a Lewy body disease patient brain, and this is how it looks in the midbrain organoid. It's not exactly the same, but it's very similar. We detect the same using immunofluorescence and other markers related with Lewy body as autophagy markers. Also, we found that in that organoids that have the triple mutation for SNCA, they are a loss of dopaminergic neurons, and also an increase of caspase-3, which is related with apoptosis. This is basically the works that I have worked. This is very basic models. It was a first screening. However, in the last 2 years, there are several studies for different models in neurological disorders. We have mostly organoids model for structural defects, infections and inflammatory disease, neurodegeneration, and neurodevelopmental and psychiatry disorders. These models have been more complex because they are evaluating not only the histology characteristics, they are evaluating functionality, transcriptomic, and other characteristics that we can understand what is the really interactions between cells in the first steps of the disease. What is the limitation of the organoids or challenge? The first is the variability or the low reproducibility, means that happen more in the brain or in the cerebral organoids or unguided organoids, because IPS have a phenomena which would produce not all the cells reprogram symmetrically, that some cells that have or keep in a different maturation stages. So this is part of the model, means that organoids, even if it's from a same patient, from a same batch, in the same well, we can get different organoids. It's like a different organoid or different brains from a same patient. That is variability. To reduce this variability, we need to produce more regional or specific organoids. Another thing is they don't contain the same proportion of different types of nerve cells as a human brain, of course, for the size. Organoids are mostly composed by neurons with a smaller proportion of glia cells, and virtually they are not oligodendrocytes. So as you know, oligodendrocytes is a kind of neural cells, very important in the neural tissue because these cells are in charge of myelinization of the axons. So it's difficult to get these oligodendrocytes and mature oligodendrocytes. There are some protocols that they try to do overexpression transcription factors, but have been complicated. And they are not completely standardized protocols for organoids. Also, we have variability in that models. So we have a necrotic core. Necrotic cores means in the center of organoid, as organoid is growing from the core or from the center to outside. Necrotic core is an area inside the organoid where the media and growth factors is not able to go, since we don't have a vascular network. So that produces apoptosis inside the organoid. And we cannot control that. For that reason, the idea is to produce vascularization in the organoids or introduce the organoid in a mouse model, for example, and mouse are able to provide this vascular system to the organoid that is named as chimeric animals. There are another disadvantage, which is the low maturity and the lack of vascularization. This model, even though it's a still infancy for large-scale application, due to the relative costly and time-consuming derivation process and the standardization. I like to talk about our current projects in the brain's research laboratory. As I told you, we need to develop organoids model more close to the human brains that involve the introduce of microglia and a blood vessel or endothelial cells. So the first project that we are working is determining the role of microglia in culture with neural organoids and develop A beta amyloid plaques and tangles. So we have a familiar Alzheimer's disease patient and sporadic patients, which we reprogramming the cells and we produce the cerebral organoid. And after 60 days, we culture these organoids with microglia from the same patient, and we left to mature these organoids that microglia can interactions with astrocytes and neurons. And we are evaluating using cell single transcriptomic and a flow cytometer if microglia, what is the microglia role in the production of tangles in A beta amyloid, and what is the proteins involved in that, to try to looking for targets for therapy, for future therapies. Another project, I will show some results about it. So we have this characterization. This characterization is more stronger. We can see in the 60 days how looks the healthy and FAD organoids, and after 120 days, how looks a more mature organoid. This is how looks the co-culture with microglia, and how looks the microglia inside the organoids, and how the microglia are able to phagocyte the A beta amyloid plates. This is I beta, sorry, I forgot to put the markers here, but this is the A beta amyloid plate. The green cells is the microglia, and we can see how this microglia phagocyte. This is the human brain, A beta amyloid plaques, and this is how it looks in the neural organoids with microglia. It's not exactly the same. We cannot mimic exactly, but we have some histological characteristics as plaques and some tau tangles in the organoids. And here is how we are starting to develop the vascularization in the organoids. We have two different models. I'm developing the vascular brain organoids, and I put in contact with the cerebral organoids to fuse each other, and the vascular cells are going through the organoid and produce the vascularization inside. We can see here some markers for endothelial cells. And at the same time, we want to introduce these organoids in the immunosuppressed mouse model to also the mouse provide this vascularization. In this moment, we want to produce the cerebral amyloid angiopathy model, which is similar to Alzheimer's disease. The difference is A beta amyloid plaques are produced in the blood vessels. For instance, we need the vessels inside the organoid. So we have patients with CAA. We are reprogramming the cells. We are using isogenic control using CRISPR-Cas9. And then the idea is to analyze the role of microglia and endothelial cells in the production of A beta amyloid plaques using cell single transcriptomic. So as you can see, the role of brain organoids in neurological research have an important role and could be an important tool in the study. So we have the in vitro human cell platforms where we can take a biopsy from a patient, produce the iPS, get organoids, and make a tissue culture with controlled environments. The future with organoids is also involves artificial intelligence. because we can put this organoid in a microelectrode. The organoids also are functional, means that they are able to produce potential actions. These neurons are completely functional. We can detect this potential action and in an electrode and transform in a digital information. So the idea is to get samples from patients, make a patient research, make genetic studies, produce IPS, organoids, transplant these organoids and in vivo models, and we can understand better disease mechanisms, or we can make a screen risk genes, understand the disease mechanisms, increase the disease understanding, improve the therapies. Also, using organoids, we can use drug testing for the same, increase disease understanding and improve therapies. And also, this is a non-invasive study. We can develop neuropathological study, patient-derived in vitro models, and patient-derived xenograft models. So thank you very much for inviting me to this talk. So I just want to say that it's not only brain organoids. Nowadays, we can develop kidney, lung, gut, and another kind of organs. Cardiac or heart organoids as well is developing right now. So nowadays, we have a very good tool to understand more the pathologies. So thank you very much. If you have any question, you can ask me in Spanish. Me pueden preguntar en español. Yo puedo traducir las preguntas. Muchísimas gracias por su atención.
Gracias, doctora Andrea, por compartirnos esta presentación tan interesante del trabajo que está desarrollando en la Universidad de Texas. Damos entonces paso a las preguntas. Vamos a iniciar aquí en el auditorio 2. De pronto, si algún asistente tiene una pregunta. Bueno, por el momento aquí en el auditorio 2 no hay preguntas. Paula, de pronto en el auditorio 1.
¿Alguien de la sala tiene preguntas? Y nos vamos preparando las personas que se encuentran por conexión. Por favor, también levanta la mano en el chat.
In English too.
Bueno, en el auditorio 2 tenemos una pregunta. Vamos a encender la cámara también, por favor.
Hola, muy buenos días, muchas gracias por la presentación. Me gustaría
Hola, disculpen. Se estaba realizando una pregunta en el auditorio 1. Profesor Andrea, ¿usted estaba escuchando la pregunta?
Sí, yo la estaba escuchando.
Perfecto, entonces de pronto hay algún tema con el audio del auditorio 2. En este momento estamos en una pregunta del auditorio 1. Te invito de pronto a que la hagamos desde acá para que también alcancen a escuchar en el auditorio 2, por favor.
Muy buenos días, entonces empiezo a repetir la pregunta y yo espero que ya haya escuchado mi introducción y pues le agradecía por esta presentación y felicitaciones en su trabajo. La pregunta específica es, bueno, en el caso mío estoy adelantando el doctorado con énfasis en genética, ¿cierto? Con una aproximación con las variantes identificadas que explican una enfermedad. En este caso, pues según lo que expuso, se ha encontrado pues como, o ha desarrollado modelos como monogénicos, es decir, en la cual parece ser que hay, según lo que explicó, una enfermedad pues como enfermedad de Alzheimer con una variante monogénica según el modelo que nos explicó en parte. pero no sé si tenga de pronto experiencia o sepa como el enfoque desde una enfermedad poligénica, es decir, desde variantes que estén interviniendo de manera, interactuando o de otras maneras en que sea un modelo complejo, si eso puede ser útil en este tipo de organoides cerebrales. Estas enfermedades que le digo, todas son cerebrales y parece ser que hay una interacción con diferentes genes que están involucrados y que pueden estar tanto de manera como epistasis o como alguna interacción génica que pueden estar produciendo.
Ok, déjame un segundo, yo traduzco la pregunta para las personas que me están escuchando de no habla hispana. She's asking that if with this model, mostly the models that have explained is most with monogenic or one genetic variant. She's asking if we can develop this model using polygenetic variants. Means I think it's different kind of genes or mutation in genes that is involved in a specific disease. My answer is yes. ¿Puedo responder en inglés o quieres que te responda en español?
Puede responder en inglés.
Okay. My answer is yes, you can. Because the advantage of a city or organoids model in general is you conserve the genetic background, even if mono or polygenetic, means that if the patient have a specific mutations, could be one or many mutation, you can conserve that mutation and you can see when the organoid is mature, is completely mature. In the depend of the gene, what is the mutation? For example, if you are studying a mutation that is expressed in the first age or when the patient is young, probably you can see in the first stage of the organoid development. But for example, in the case of neurodegenerative diseases, this is more related with aging. We only can see these genetic profiles when the organoids are aging as well. So we need to induce aging in the organoid actually to see these characteristics. But mostly you can use, for sure, you can use many genetic background and you can see in the organoid, and also even using sporadic. For example, in patients with sporadic Alzheimer's disease, you can see some of the characteristics in the organoids. Remember that sporadic diseases is not only for genetic factors. It's only for environmental factors and makes more complicated to extrapolate in models. However, the last studies have shown, mostly using cell single transcriptomic, that we can conserve in the organoid these characteristics and then see in the cells after differentiation. In embryonic stages, no. At least that you are studying a developmental disease or diseases related with development. But if disease is related more with aging, you will see this characteristic when the organoid old. More, and means that you need to get organoids than more than 4 or 5 months growing.
Thank you.
I don't know if I answered your questions.
Pasemos a la pregunta de la sala 2. Daniel.
Hola, buenos días de nuevo. Gracias por la presentación. Mi pregunta va más enfocada como en esa limitante que hay como en la parte vascular de los organoides y que se genera pues ese núcleo necrótico, núcleo de muerte. ¿Cómo ves en el futuro la posibilidad, por ejemplo, de generar organoides con una vascularización como para poder suplir esas limitantes y hacerlos un poco más, llamémoslo, más reales a un modelo pues animal o ya un ente más biológico o más fisiológico?
Yeah, sorry, he's asking about the disadvantage of organoid, which is the necrotic core. Yeah, this is a big issue in the organoid model. We have two options right now, but there are another proposal. I will explain the two options. One is make assembloid vascular organoids. I have explained that we are not able only to produce brain organoids. Mostly you can produce or mimic different organs of the body, include vascular organoids. What is a vascular organoid? It's an organoid only with a vascular network. And if we fused both the cerebral one with vascular, the cells are able to move or go through the organoid and proliferate the endothelial cells and make vascular networks. Actually, there are two models in that, and we can see vascularization inside. What is the problem with that? We can get vascular network, but we don't have a heart. Means that there are no one that pumping blood or pumping media in the organoid. So we can get the vascular shape inside the organoid, but even though we cannot introduce the media inside the organoid. So even we can get the vascular thing, or the vascular cells and the vascular network, but we don't have media inside or in the core of the organoid. That is a big problem. Now, or currently, there are two methods. One, put the organoid in a special bioreactors that they mimics as a heart and pumping the media inside the organoid. Another one is make chimeric animals. What means chimeric animals? Introduce the organoid in an animal, in a mouse, for example, and mouse provide the vascular system. So imagine that it's like introduce a tumor or introduce a tissue it's a human tissue in a mouse, and all the vascular system go through the organoid and embed in the organoid and produce the vascularization. This is the best way, actually, in nowadays. And there are some reports that show that both human and mouse cells start to interact each other and start to produce a new tissue that is named chimeric animals. This is another way. The last research that tried to improve this disadvantage is use 3D or develop using 3D printers. So they are produce the vascularization using 3D printers and then put in contact with the. So this is very new. This is still in a study, but for now, we have just these two options: make assembloids with vascular organoids or introduce or transplant in an animal. Or for analysis as well. In our case, for example, in our previous work, we skip the necrotic area. We don't analyze this area, and just we analyze the outside area of the organoid. So the area that grows the cells. The cells always grow from inside to outside because this is the way of neurodevelopment. So this is other thing we want to use another technique that is a cell single transcriptomic, but is localized. We are using the transcriptomic directly in the tissue in a slide. And we can analyze the transcriptomic of specific regions of the organoid, and we can skip this area.
Muchas gracias.
No sé si resolví tu pregunta.
Sí, profe, muchas gracias.
Bueno, con gusto.
¿Alguna otra persona del auditorio, sala 1? Profesor, lo invito para que nos alcancen a escuchar en el auditorio 2, no sé por qué el inalámbrico no está captando.
Thank you, doctor. Nice talk. I have a question related with the use of organoids in blood-barrier models. I don't know if you have any experience that you can share with us.
With blood-brain barrier, no, we're working on it actually, because previous my project was develop like basic models. Mean in the beginning we didn't know if Alzheimer organoid were able to produce a beta amyloid plaques, which is a classic histology characteristic of the disease. We didn't know, but we saw that, yes, the organoid is able to produce that. Then we need to move to something more robust. So produce the blood-brain barrier is complicated. Our first idea is try to do the assembloids between vascular organoids and cerebral organoids. It's a complicated technique because but each organoid that needs a specific small molecules or growth factors to differentiate, and then try to fuse and make the gradients of growth factors in both is complicated. So you need to think in some point, what is more important, to preserve the neural cells or preserve the vascular system? So you cannot just mix neural growth factors and vascular growth factors at the same time. In theory, when you have organoid cells, each other send paracrine signals, mean that send growth factors specifically for the other cells that is able to survive. But in vitro sometimes works, sometimes don't. This is a big challenge. So produce the blood-brain barrier. Yeah, maybe it could be possible in the future. First, we wanted to produce a vascular network. If we are able to produce a vascular network inside the organoid, probably we can move to blood-brain barrier. So let's see. Only I let you know if we can reach that.
Muchas gracias, profesora, por esta presentación. A todos los que nos acompañaron también por este espacio de preguntas. Por razones de tiempo, vamos a dar en este momento ya por finalizada también el momento de preguntas. Le agradecemos nuevamente a la profesora Andrea Becerra, quien es egresada de nuestro programa, por compartir con nosotros esta presentación, que también será ubicada luego en nuestro canal de YouTube para que podamos revivirla en caso de que pronto nos perdimos alguna parte. Muchas gracias, profesora.
Muchísimas gracias, y simplemente quería invitarlos a todos. Esto es nuestro grupo de investigación. Nos pueden hacer el scan del QR, del código Q, ver nuestro grupo. Nosotros tenemos proyectos abiertos, recibimos postdoc, y si alguno de ustedes quiere escribirme y si tiene preguntas, este es mi correo electrónico. Paula lo puede compartir sin ningún problema, y con mucho gusto estoy aquí para colaborarles y para poder hablar del tema. Y bueno, estamos interesados obviamente en que ustedes también les guste el tema y puedan venir y aprender también. Aquí en la universidad estamos abiertos a recibirlos.
Muchísimas gracias, profesora, y a todos los que nos acompañaron a través de la conexión y en el auditorio.
Gracias.
Bueno, están hablando en el auditorio 1, aquí no estamos escuchando en auditorio 2.